And our last question on this paper is question 7, which is about antibiotics that are drugs that are used to treat bacterial infections. We also talk about the fact that mutations in bacteria can produce new strains or types of bacteria, and these strains are resistant to antibiotics. And 7.1 asks us, where do mutations happen in the bacterial cell? This happens in the DNA of the bacteria, which is in the plasmid, or we could say it's in the chromosomes. We know, of course, that bacteria do not have nuclei, so the nucleus would not be an acceptable answer for this one. If we have a look at the next question, next part of the question, we're looking at Staphylococcus aureus, and we've put it into a Petri dish. We've got some discs of paper that we've soaked in antibiotic solutions, put them in there, and we've incubated it at 37 degrees for 24 hours, and these are the results. So we've got two discs here that have no zones of inhibition near them, which means that there's bacteria growing right up to the circle itself, and then we have 3 others where there is a clear space around each disc. The scientists concluded that S. aureus is resistant to antibiotics C and E, give evidence for this conclusion. So there is no clear area, no zone of inhibition around discs C and E. So this tells me that no bacteria were killed, so no bacteria were killed. 7.3, the scientists later discovered that S. aureus is not resistant. How would they have done that? So I think the easiest thing they could have done would be to increase the concentration of the antibiotic, and then you would have seen a clear area seen around the disc. Moving on to 7.4, so we have some information about an experiment. So we've got 4 treatments. So broken bones are sometimes repaired using a metal implant. This can be infected and it's treated with long-term antibiotics. This has led to antibiotic resistance in some bacteria, so we're looking at alternative treatments. They suggest that you could use stem cells, which are undifferentiated cells from either bone marrow or fat tissue that have antimicrobial properties that destroy bacteria, etc. So they investigated 4 treatments on an area of infection, and each treatment was injected into the area around the implant. So you've got 4 different treatments here. It was tested on 5 patients for 7 days, and then they calculated the area of infection over the total tissue area. Independent variable, so this is what is changed, and they clearly changed the treatment, so the treatment injected around the implant was the correct answer here. Suggest one advantage of using stem cells from fat tissue rather than bone marrow. This relatively straightforward. You've got the idea of if you're taking it from bone marrow, you have to drill into the bone and harvest it that way, which has obviously problems associated with it, whereas harvesting from fat is a lot easier. So it's easier to obtain, easier to get. Or you could have said it is less invasive. You've got fewer side effects, it's less painful. 7.6, how did the antibiotic enter the stem cells from the solution? It would have had to have been by diffusion, because the concentration of antibiotics inside the cell was lower than outside the cell. And diffusion is where particles move from a region of a high concentration to a low concentration. So that matches with this one. What was the range of results for the treatments with stem cells only? So we're looking here. So this is the range we're interested in, and our lowest one was 0.9 and our highest was 0.31. So 0.09 all the way up to 0.31. So your range is the smallest to the highest. Our last question. Somebody looked at the results. They concluded that the injection of stem cells containing the antibiotics should be used to treat all implant-related infections, and we need to evaluate it. So we need to come up with some pros, some cons, and our thoughts and feelings, so our judgment. So for me, the pros are that the stem cells had the smallest area of infection. They had the smallest range of results. And there was no overlap with the unreactive solution. So if we go back to here, say no overlap with the unreactive solution at all the stem cells containing the antibiotic. Cons. So why might they not agree with this? So only tested on 5 patients, so very, very small sample size. Similar to other treatments, so similar results to other treatments. And so there were lots of variables that were not controlled, so age or health, not controlled. Okay, thank you very much.